Good afternoon, sir.
Hello, Ramnath. Good afternoon.
How are you?
Yeah, fine.
तुमचा आवाज कमी येतो सर.
Pardon?
Yes, sir. Yes, sir.
थोड़ा प्रॉब्लम आ रहा है.
Beloved sir, we can starting within 5 minutes. We are wait for the 5 minutes.
Okay, fine.
काम करतो यांचा रिस्टार्ट मारा.
हेडफोनची गरज नाही ना. ठीक आहे. फुल आहे ना व्हॉल्यूम. Good afternoon, everyone. Sorry for the late, first of all. Because of some internet issue, I was unable to connect. So I connected with the Ramnath sir's laptop. And sorry, Belavdi sir, for keeping you waiting. Okay. Thank you, sir. Good afternoon, everyone. It's an honor to have you all here for this last session of the lecture series. Now, first of all, I'll welcome you all, and I welcome Belavdi sir also on the behalf of the principal of our college, Dr. B.B. Gaikwad sir, even the HOD, Dr. Vikram Kakurte sir, and organizing committee members. I welcome you all. Dear participants, today we have the eminent speaker with us, Dr. Vasuki Belavdi sir, and let me introduce him first of all. So Dr. Vasuki sir is a former head and ICAR eminent scientist at Department of Entomology, Bangalore. Even the Dr. Belavdi sir also did his PhD in agricultural entomology, which is quite interesting subject. He is even the recipient of many honors and awards. He was working as a visiting faculty, visiting fellow at University of Southampton at UK. Even he is a fellow of the Royal Entomological Society, London since 1995. Sir is also the awardee of Swami Memorial Award, Narasimha Swami Memorial Award in 1998. He is also recipient of the Mehta Memorial Award in 2002. Even he also worked as a consultant for the global pollination project during the year 2010 to 12. Even he is a member of midterm evaluation term of the global pollination project for the UNEP since 2012. Sir is also received the Best Teacher Award from ICAR during 2013. He is a member research advisory committee at ICAR National Research Center for the Orchids, which is at the Sikkim. Even Sir is also the member of the review team since 2019 at AICRP on the bees, honeybees, and the pollinators. Even apart from these huge honors and awards, he has a great contribution in teaching. He used to teach entomology course for the BSc and MSc agriculture. Even his great contribution is there in guiding PhD students at the Bangalore University. Even he has a great hand in research. He did almost 9 outstanding projects which were funded by the various national institutes like DST, DBT. Even apart from that, Sir is very interested in the research which is related to the pollination ecology as well as the taxonomy of solitary bees. And Sir has also guided near about 15 MSc students for their master's project as well as 14 PhD students for their PhD dissertation. Even Sir has a great contribution in research as well as he has a very big number of research publications. Sir has published 124 research papers in various national as well as international journals. Even he has published 12 popular articles too. And apart from that, Sir has published 2 books as well as many extension folders, even some bulletins, etc. Apart from that, Sir has delivered more than 80 guest lectures to reputed universities, colleges, as well as institutions. So dear participants, we have such an eminent speaker with us. And now let's hear him. Over to you, sir.
Yeah, thank you very much for introducing me. I will now share my presentation.
Yes, sir.
Yes, it is open.
Okay, is it visible? Yeah.
Yes, it is visible.
Okay. Okay, friends, I'll be speaking today on bee diversity, taxonomy, and conservation. Actually, I have 3 parts in this talk. One is about the evolution of bees, evolution of pollinators, and plants. Okay, plant-pollinator interaction, how it started. We'll try to understand that. And then a little bit about the taxonomy of bees, how many kinds of bees are there, how to identify them. And I will also give 2 examples to show why bees are important as pollinators and why we need to conserve them. Okay, with this background, I'm going to start now. Now, coming to the evolution of the plant-pollinator interactions, you know that flowering plants appeared about 130 to 160 million years ago. And in fact, Dave Goulson, in one of his books published in 2014, which is A Sting in the Tail, he says that sex has always been difficult for plants because they cannot move. And if one cannot move, then finding a suitable partner for reproduction becomes almost impossible. So this is a very famous statement by Goulson in his book. And of course, there was self-pollination, but there are disadvantages with self-pollination because the main problem with self-pollination is that the population will have very low or very little variability. And so the populations may succumb to any biotic or abiotic stresses, and there is no question of survival of the fittest because every individual in the population will be the same. Okay, so the entire species may get wiped out if it entirely depends on self-pollination. And that is one of the reasons why plants slowly resorted to cross-pollination. So they have to invariably depend on sex facilitators because they are incapable of moving. Now, about 135 million years ago, plants discovered wind pollination. Wind pollination also had its own disadvantages, or it has certain disadvantages, because a wind pollinated plant has to produce very large number of pollen grains because the success rate is very low. The success rate, in the sense, the chances of the pollen reaching the stigma of the right flower of the right plant is very, very low. And so the plant has to produce very large number of pollen grains, and so majority of the pollen goes waste. So a lot of energy is wasted with wind pollination. Now, between 130 and 120 million years ago, some insects discovered that pollen grains are edible and nutritious. So they started feeding on them. Some winged insects soon began feeding on the pollen, and they became specialists in eating them. So there is fossil evidence to show this, that Cretaceous tumbling flower beetle existed, which was actually feeding on pollen. Now, flying from plant to plant in search of their food, they accidentally carried pollen grains on their bodies, trapped again amongst the hairs or in the joints between their segments. And when by chance, accidentally, some pollen grains fell off the insect onto the female parts of another flower, that flower was pollinated. And this was the beginning of cross-pollination. Now, there are even today the cycads, which were the earliest plants to get pollinated by beetles, they are still pollinated by beetles. Now, although much of the pollen was consumed by the insects, this was still a vast improvement over their dependence on wind. So plants soon learned the advantages of insects visiting their flowers. So there is a lot of saving of pollen if they depend on insects. So this is what the plants learn. And the plants were benefited since pollination was assured, but still there was a problem. Because all the early flowers were greenish in color, you know that the flowers are modifications of the foliage or the leaves, they were also greenish in color. So the early pollinators had difficulty in locating flowers. In order to attract insects, plants had to get better and advertise their flowers with different colors than the surrounding vegetation. Probably it took several million years for this to happen. A mutation probably occurred which resulted in a different colored flower. So the earliest flowers which were recorded were the early lilies and magnolias, which are still there, were the first plants to evolve petals conspicuously white against the greenish vegetation. Thus began a selection for flower color. Okay, so the natural selection acted on those plants which had different colors to attract different pollinators. Now, the second trick that the plants resorted to was offering a sugar-rich nectar as an extra reward, possibly again to save pollen. So with this new reliable means of pollination, insect-pollinated plants became enormously successful and diversified. And this, in fact, they also resulted in specialization in insects. Moths and butterflies and some flies evolved long tubular mouthparts to suck the nectar, and thereby they also transferred pollen, and which resulted in cross-pollination. And the most specialized and successful group to emerge were the bees, the masters of gathering nectar and pollen to this day. And the first true bees appeared between 132 and 113 million years ago. So the bees are more than 100 million years old. Competition for pollinators actually resulted in selection for brighter colors, patterns, elaborate shapes, and variations in quantity and quality of rewards offered by flowers. Today, if you go out of any building into a garden or even into the forest or a countryside, you see plenty of plants, and each plant with a different kind of flower. So all that is a result of competition for pollinators. This led to a sudden increase in speciation among plants around 100 million years ago. And this, in fact, is referred to as pollinator-mediated speciation in flowering plants. Okay? So if we have so many kinds of flowers, so many kinds of plants with different kinds of flowers, it's thanks to the pollinators, which actually acted as the selective force. So why pollination is important? Pollination is an essential service to crops and wild plants. Pollinators are vectors for genetic exchange. The variability that you see among plants is mainly because of pollinators. They bring the genetic material from other sources, from different plants, and they are responsible for the variability that you see within a population. And it is essential in obligate mutual fig and the fig wasps. There are several species of figs. There are about 800 species of figs in the world, and there are an equal number of fig wasps. There is a one-to-one relationship between the fig and the fig wasps. So there is an obligate mutualism. Then they also provide other ecosystem services, like in the wild, in a forest, there are plenty of birds and animals which depend on fruits and pods that are set in the wild. And all that food is the result of pollination by insects, by these bees, butterflies, beetles, and many other pollinators. And they also have biodiversity value. If you have a high biodiversity today, it is thanks to pollinators, which actually brought in so many species of plants, and so many species of plants resulted in the arrival of several species of animals that depend on them. Say the bird diversity, animal diversity, plant diversity, all these, I would say, is a result of the work of pollinators. Now, we would not have many of these if the pollinators were not there. Now, why only bee? The word bee is almost synonymous with pollinator. And when we hear the word bee, we think only about honeybees. But there are nearly 20,507 species of bees in the world, and there may be more than 25,000 species in the world. And in India, we have 727 species of bees, which is just 3.5% of the total number of bees recorded in the world. And there may be many more species. Bees actually belong to the order Hymenoptera under the superfamily Apoidea. Now I'll come to a little bit about the taxonomy of bees. Now the superfamily Apoidea has two groups, Speciformis and Apiformis. Speciformis includes all the species wasps or spicoid wasps, which are predaceous. They feed on other insects. Okay. Now Apiformis includes all bees. The main difference between Speciformis and Apiformis is with the structure of the hairs that are present on the body. The hairs that are present on the bees are branched. Okay, they have branched hairs. Okay, branched setae. Whereas the Speciformis or the spicoid wasps either do not have hairs, or if they have hairs, the hairs are not branched. So this is a very simple way of separating out a specific wasp from a bee. Now, coming to bees of India, there are in the world there are 7 families of bees: Antonidae, Apidae, Collectidae, Halictidae, Megachilidae, Melitidae, and Stenotritidae. In India, we do not have any representation for Stenotritidae. We have representatives for the other 6 families, and the most species or the species with the highest number of genera are in India is Megachilidae with 27 genera. Apidae has 24 genera, and the total number of genera in India is 70, which represents about 14% of the total genera of bees in the world, which is 528. Now, if you look at the number of species, the family Megachilidae is has the highest number of species, with 229 species. Apidae has 223 species. Halictidae has 219 species, and the others have lower number of species. So the total number of species is 727, as I just referred to you. So coming to the characters of these bees, how we can separate out these families, bees can be grouped into 2 large groups as long-tongued bees and short-tongued bees. The long-tongued bees are referred so because the labial pulp has 4 segments. The first 2 segments of the labial pulp are longer, significantly longer compared to the remaining 2 segments. So the first 2 segments are very long, and the glossa is slightly expanded. And Megachilidae and Apidae are the long-tongued bees. Now the short-tongued bees, the character is the labial pulp here also is 4-segmented, but all segments are small and equal, and the glossa is pointed. So the remaining 4 families, Collectidae, Antonidae, Halictidae, and Melitidae, are the short-tongued bees. So the long-tongued bees are Apidae and Megachilidae. Short-tongued bees are Collectidae, Antonidae, Halictidae, and Melitidae. Now, coming to the identification characters of these families, Apidae and Megachilidae, they can be very easily separated or differentiated by the presence of hairs on the hind leg to carry the pollen. The hairs are present or for carrying pollen on the hind leg, which is called corbiculum. Whereas in case of megachilids, they have hairs on the ventral aspect of abdomen. Okay, so this is referred to as this is the important characteristic feet. They are called as the scopae. The scopae are present on the ventral aspect of the abdomen for carrying pollen, whereas scopae are present on the hind legs in apidae. Okay, so that is the main difference. Another important difference is there are three submarginal cells in the forewings. These are the three submarginal cells in the forewing in apidae, whereas in megachilidae, they have only two submarginal cells in the forewing. So these are the two major differences between apidae and megachilidae. Then coming to collectidae and antennidae, the glossa is bilobed in collectidae. It is split like this, whereas in antennidae, it is pointed. The glossa is pointed. And coming to halictidae and mellictidae, the basal vein is curved or arched in halictidae, whereas the basal vein in the forewing is almost straight in mellictidae. So these are the simplest characters which can be used for identifying the families of these bees. You can very easily identify them. Now coming to the distribution of species and the genera, you see that when in the family Apidae, we have 223 species in 24 genera, and of this, Bombus is the highest, has the highest number of species with 53 species. Bombus are the bumblebees. Bumblebees have the highest number of species. This is followed by Xylocopa or the carpenter bees, which have nearly 28 species. And the next one is Ceratina and Nomada and Amegilla. So all the others have very small number of species. In fact, when we are talking about bees, we say that we talk only about honeybees. In fact, there are only about 6 or 7 species of honeybees in India. Okay, in the world, there are only about 12 species of honeybees, which belong to the genus Apis. Now, the family Halictidae has 219 species, and the most species is Lasioglossum. The genus Lasioglossum has the highest number of species. This is followed by Lipotrichus with 45 species. Now, all these are solitary bees again. Megachilidae again has 229 species in 27 genera, and megachile, which includes the leaf cutter bees. These are referred to as leaf cutter bees. I'll talk more about this when I mention about an example. Okay, with 93 species in India. This is the highest number of species. Now, these are excellent pollinators, many crops. Then coming to the remaining 3 families, which are represented with lower number of species. Antennidae is represented by a single genus, Antenna, with 21 species. Collectidae has 31 species in 2 genera, Colletus and Hyalias. And Melitidae has 4 species in under 2 genera, Dasypoda and Melita. So this is in short about the diversity of bees that we have in India. We have 727 species of bees in India, and unfortunately, we concentrate only on 1 or 2 species of honeybees, okay, when we have so many species which are excellent pollinators of our crops. Now I'll take 2 examples to explain why these bees are important as pollinators. One, I am taking a non-apis bee, which is not a honeybee, which is pollinated not by a honeybee, and the other example is pollinated by honeybees, okay. So one is pigeon bee or red gram, and the other is muskmelon. So, and I will also make an attempt to estimate the value of the pollination service. So in fact, if we have to conserve the pollinators, we should know what is the value of the pollinator. So that's the reason to justify why we need to conserve bees, we need to know the value of the pollinators. Now, coming to pigeon bee, you can stop me at any time if you have any questions, okay? So,
yes, sir. Yes, sir.
Yeah. Pigeon bee is Gajanus gajan. Now, this is supposed to have originated in southern or eastern India, and you know that it is one of the major pulse crops in India. And I don't think there is any home in India which does not use pigeon bee. Okay, tur is one of the major, pulses consumed by Indians. Okay, it is cultivated in over 50 tropical and subtropical countries around the world. and it has a very high nutritional value, major protein source for vegetarians. Food and nutritional security for developing countries depends on this crop. Now, in India, it is cultivated over an area of 4.23 million hectares, and the annual production is 3.75 million tons. Now, the productivity is 806 kgs per hectare, but the potentiality for production is 2,500 kg per hectare. So we have not even reached half of the potentiality of the production of this crop. Now, the low productivity has been recorded, and there is also a potential to enhance. Let's see how we can enhance the productivity. Now, the reasons for low productivity often that is given are insect pests and diseases. There are many insect pests which bother the crop, and there are also diseases which bother the crop. So most often, agricultural scientists are bent upon telling that these two are the major factors for low productivity of any crop. Now, there can be another possible reason. One is inadequate knowledge of pollination. We have not understood the pollinator requirement or pollination requirement of the crop properly. It is considered as often cross-pollinated, and if you look at old literature, it says that the cross-pollination ranges from 3 to 40%. And if you go and then see any expert in the field, okay, any scientist who is working on red gram, the pot set, he says, is just around 30%. Now, if you go to any farmer and if you do yourself an analysis of the number of flowers that are produced and the number of pots actually set, you will actually see that the pot set is only about 20%. Now, the question is there a pollinator deficit? Have we not seen what is the pollinator? Are we not thinking about conserving or enhancing the population of those pollinators? So that is the question. So before understanding or before studying the pollination biology of any crop, it is important to understand the floral biology of the crop. Okay, so here, floral biology of pigeon pea, if you look at, it is self-compatible. So that means to say there is a possibility of self-pollination to a certain extent. But it is capable of setting seeds by self-pollination, but benefits from cross-pollination. If there is a pollinator, if it benefits from cross-pollination. What is the benefit that it is getting? It is getting the pollen from outside, from other plants, so that the variability is higher. So the better genes are coming, and better, we say the pollen is coming, and then the quality of the seeds and the quality of the produce will be better. So the flower structure, if you look at, it is a typical papilionaceous flower with standard wing and keel petal. Now, the interesting thing here is all the reproductive parts, that is the stamens or the anther lobes and the pistil, style, and the stigma are hidden beneath the keel petal. Okay, so it is not exposed at all. It is beneath here. Now, supposing if a pollinator wants to take nectar also is produced in these flowers. If it wants to take the nectar, it has to see, and if it is to pollinate the flower, it has to see that the reproductive structures come out of the keel petal. We will discuss about that. So the stamens are 10 diadelphous. That means they are in two groups, one group of 9 and fused into a column and one free. And the ovary is superior, subsessile, flat dorsoventrally, unilocular with 5 ovules. Stigma is capitate. Nectaries are there at the base of the style. And if you look at the pollen-ovule ratio, it is 1,000 to 1. There are nearly 5,000 pollen grains that are produced in a single flower. There are, as I said, there are 5 ovules. So 5,000 pollen grains are there, and then 5 ovules are there. So the ratio is 1,000 to 1. Now, the flowers are visited by 18 species of insects, including 12 species of bees. Now, first we wanted to look at whether these flower visitors help in any way to increase the pot set. So what we did was we had sets of plants which we caged, about 25, a set of 25 plants were completely caged, and another set was left open for pollination for flower visitors. Now, interestingly, we found that there was a significant increase in pot set in plants that were left open compared to the caged plants. It indicates that the caged plants also are capable of setting pods, but because of the self-pollination, they are self-compatible, and the self-pollination results in the setting of less than about 20%, about 17 or 18% of the flowers are setting into pods. Whereas when you are leaving it open, it is about 30 to 35%. Okay, so there is a significant increase. So if the flower visitors are helping, what are these flower visitors? So we looked at the bees which are visiting, and we found that there are about 9 species of Apidae, bees which belong to Apidae. Like there are 3 species of Apis dorsata, florea, and serrana, the honeybees. Then about 4 species of carpenter bees, Xylocopa, Latypus, Amethystinae, then Estuans, and an unidentified species. Then Siratina, 1 species, Tetragotilla, 1 species, and there are 3 species of Megachilidae, or the leafcutter bees. Now, to look at the significant flower visitors, which are of these 12 flower visitors, which are the ones which visited most frequently? So we found that there are about 6 species of bees which are visiting more often, and of these, we found that Megachile lanata and Apis florea are more frequent visitors. Highest number of visits, highest percentage of visits were by these 2 species, followed by Xylocopa amethystina. Now we wanted to look at which of these 3 species are good pollinators. Okay, so we did some experiments, controlled experiments, in which we allowed visits by only Megachile lanata, only by Xylocopa, only by Apis florea, and we compared that with a set of another set of plants which was left for open pollination and which was closed for preventing pollinators from visiting. And we found that the percent pod set in those plants which are visited by Xylocopa and Apis florea were not significantly different from those plants which were not visited by any bees. Okay, which indicates that these are not actually contributing anything to the pollination, but these are also self-pollinated, like it happened in no visits. But the number of flowers which were visited by Megachile lanata, the percent setting was almost equal to the open pollination. Okay, which shows that Megachile lanata could be the major pollinator. But there may be some other reasons also which we wanted to look at. So this is Megachile lanata, which I said is a leafcutter bee. It actually is a solitary bee which constructs nests in existing holes, or holes in the trees or in the walls. If there are holes, they come and then construct nests, and they line the cells with bits of these leaves that they cut from different plants, and then they store in the cells a mixture of pollen and nectar on which they lay an egg, and the egg hatches and the larva feeds on the mixture of pollen and nectar and then develops into an adult. So a single female may have several cells within a nest, and then it can also build several nests nearby. Okay, so that is the brief biology about Megachile species. Now we found that when we looked at the behavior of these bees, Megachile lanata, Xylocopa, and Apis florea, we found that the searching time that is taken by Megachile is much, much shorter compared to Xylocopa and Apis florea for finding the flower. The searching time was significantly lower here, and the number of flowers visited per minute is much higher with Megachile lanata compared to Xylocopa. Because Xylocopa spends more time, Xylocopa and Apis florea spend more time in searching for the flower, the number of flowers that they visit will be usually less. Whereas Megachile lanata is much faster in locating the flower, searching, and then it knows how to take the nectar. And so it spends least time searching for flowers and visits maximum number of flowers per unit time. Now, there is also another advantage with Megachile lanata. It also knows where to sit, how to press the wing petals, and how to take out when it presses the wing petals, the entire anther lobes come out, and then it brushes against the setae that it has on the ventral aspect of the body. So this clearly shows that Megachile is made for pollinating pigeon pea or pigeon pea-like plants. okay, or papilionaceous or leguminous plants. Okay, so it visits and then it presses the keel petals, wing petals, and the anther lobes and the style and the stigma come out, and then they brush against the hairs that are there, scopae that are there on the ventral aspect of the abdomen, and it either deposits the pollen, or the pollen is deposited on the stigma, and the pollination is affected. So megachyle is better equipped for pollination as far as pigeon bee is concerned. So this was the first proof that we have. Then, can pod set be improved by increasing leafcutter bee population? So this is the next question that we wanted to tackle. So we selected a large plot with about 4,800 plants, and placed about 20 trap nests. The trap nests, what we made was we used ipomia reeds, cut them into about 6 to 8 inches long pieces. Ipomia reeds are usually hollow. When you dry it, is completely hollow tube-like thing. And then we placed these reeds inside a PVC pipe, and then hung these things in the field. So each trap nest contained about 12 to 15 centimeter long hollow reeds. The total number of reeds that we placed was about 720. So each one is a location, a site for a megachylid to come and then nest, okay, for nesting the megachiles. So in the same plot, about 40 plants were caged to exclude flower visitors. And we also had a control plot which was left for open pollination, which was about 500 meters away. Okay, so we wanted to compare when a plot which is completely open pollinated with a natural population, a plot where we have tried to increase the number of, say, the nesting sites, and then increase the population of these bees. In fact, we found that out of the 720 reeds, nearly 150 or so were occupied. That means so many bees were active, okay, compared to other places. So observations were recorded on the pod set in all these three situations. Now, interestingly, we found that in the open pollination, the setting was around 30 to 35%, whereas when in a plot where we had increased the population of leafcutter bees, the setting was more than 60%. So providing nesting sites increased the leafcutter bee population, and increased the activity of leafcutter bee almost doubled the pod set. So this is a very clear indication that we can actually increase the productivity of red gram if we conserve the populations of leafcutter bees, or improve the population of leafcutter bees. So though pigeon bee is self-compatible, it is benefited by visits by leafcutter bees. The flower structure appears to have evolved for pollination by megachyle species, and by increasing the nesting sites for leafcutter bees, we can in fact increase the pod set significantly. And there is a need to conserve leafcutter bees and their nesting sites. Now, in fact, in many places, there is a concept called bee hotels. So the bee hotels are nothing but we can actually construct this kind of small structures, small boxes with many tube-like structures for the leafcutter bees to nest. In addition to leafcutter bees, many other bees also make nests in these kind of hollows, and that increases the population of pollinators in any given area. So this, I think, we have to make farmers aware of this, and then improve this understanding. Shall I move on to the next one? Okay. So the next example that I'm taking is pollination in muskmelon. So muskmelon is Cucumis mellow. Now, the most important thing about muskmelon is that it is anthromonaceous. That means there are two kinds of flowers, hermaphrodite flowers and staminate flowers. So both kinds of flowers are there in this. Now, the question is, when the hermaphrodite flowers are there, why it has to produce staminate flowers? You know that hermaphrodite flower means it has pistil, and also it has ovaries, and also it has stamens. Okay, so when the stamens are there, when the pollen is produced in the hermaphrodite flowers, why it has to produce staminate flowers also? So that is the major question that we wanted to answer. Now, we found that the flowers of muskmelon are visited by several bees, including 3 species of Apis, honeybees, mainly Apis florea and Apis cerana, were visiting, in addition to many other solitary bees. Now, honeybees, especially Apis cerana and florea, are the most frequent visitors of muskmelon. Now, the questions that we asked, as I said, were why staminate flowers? When hermaphrodite flowers are there, why the plant should produce staminate flowers? Are pollen from hermaphrodite flowers and those from staminate flowers the same? Is there any difference between the pollen? If both the flowers are producing pollen, are they same or are they different in structure, viability, fertility, etc.? Now, if they are not different, what is the purpose of the male part in the hermaphrodite flower? So these are some of the basic questions that we wanted to answer. Now, if you take the count of the number of staminate and hermaphrodite flowers and work out the ratio, you find in many cucurbits, you'll find this, the number of staminate flowers is much, much higher compared to the number of hermaphrodite or female flowers. Now, the ratio is 18:1 in this case. Now, when we flowers, so in hermaphrodite flowers, the number of pollen grains produced is less. In staminate flowers, the number of pollen produced is nearly 5,000, okay, per flower. And when you look at the viability, there is slight difference between the viability. Viability is just germination. When you put the pollen for germination, there are germination tests can be done very easily. Okay, it is simply one of the simplest way of working out the germination test or doing the germination test is putting this pollen in 10% sugar solution, okay, sucrose solution. So it germinates. So when you see that, there are many other chemicals also which are used, and that there are more complicated ways of working out germination tests also. But when you do the germination test, a proper germination test, the staminate flowers have slightly higher percent germination compared to hermaphrodite flowers. But another important difference that we noticed was that after germination, the pollen from the hermaphrodite flower does not grow. The pollen tube does not grow further. Okay, within about 1.1 micrometers, it stops growing, whereas the pollen tube of the pollen from staminate flower, it continues to grow. It goes beyond the reach of the first ovule or the presence of the ovule. The stigma to the first ovule length is about 6.5 micrometers. So you can say that it actually exceeds that, and then it can reach the ovule and then fertilize. So this gives an indication that the pollen from hermaphrodite flowers are in fact not involved in reproduction. They are not actually fertilizing the ovule, whereas it is only the pollen from the staminate flower that is fertilizing the ovules. So it is important for the plant to produce staminate flowers. But the question is why it should have in the hermaphrodite flower the male part. Okay, pollen from hermaphrodite versus we did some experiments also, crossing experiments, hand pollination experiments, and it was proved that pollen from staminate flowers only was able to set the seeds or the fruits, whereas the pollen from hermaphrodite flower, whether it is selfing or bagging or to another flower, it did not actually set any fruit. So the pollen from the hermaphrodite flower was not useful for fertilization or reproduction of the plant. So hermaphrodite flowers that were pollinated using pollen from the same flower or from other hermaphrodite flowers did not result in fruit set. On the other hand, with pollen from staminate flowers resulted in 100% fruit set. So the question is, what's the purpose of pollen in hermaphrodite flowers? So why it should continue to produce pollen in hermaphrodite flowers? So for this, we did some emasculation studies. So in the emasculation study is nothing but you just remove the stamens, okay, the male part in the hermaphrodite flowers. So we had a set of flowers, but a set of 30 flowers which are normal hermaphrodite flowers where the stamen is intact, the male part is intact, and for another set of 30 flowers, we emasculated. That means the male part is completely removed. And when we looked at the percent fruit set, we found that there is a reduction in the fruit set. There is slight reduction in the fruit set. It is 100%, it is 80%. Now, when we looked at the number of seeds that are produced, you'll see that there is a significant reduction in the emasculated hermaphrodite flowers when when the male structure is not there, the seed set is very, very low. So why this difference in fruit set and seed number is the next question. Then we looked at the pollinators. Apis cerana and Apis florea both are excellent pollinators of this plant, this muskmelon. And in both the hermaphrodite and in normal hermaphrodite and emasculated hermaphrodite flowers, when we observed for the time that is spent by the pollinators, we found that the bees actually spent significantly longer time in normal hermaphrodite flowers compared to the emasculated hermaphrodite flowers, which means that in the emasculated hermaphrodite flower, it is spending very little time because it is not getting pollen, it is not finding any pollen, and probably in the normal hermaphrodite flower, it is actually searching for pollen, and then in the meantime, it also pollinates. Okay, it also deposits pollen that it has brought from the staminate flowers. Now, the number of pollen deposited also is significantly different between the normal hermaphrodite flower and emasculated hermaphrodite flower. So this is this very clearly indicated that there is some purpose for the pollen that is produced in the hermaphrodite flowers. So in the normal flowers, we spent more time as they also collected the pollen, and probably pollinators spent less time on emasculated flowers because they were not getting any pollen. And probably this pollen was actually the hermaphrodite flower is giving the pollen to retain the pollinators, which the pollen can be taken by the bees as food. Okay, the food, in fact, it is very interesting if we have to collect the pollen grains from the hive and then see which kind of pollen is coming more, which we have not done yet. So muskmelon requires bees, including wild bee species, for pollination. And in muskmelon, the male part in hermaphrodite flowers helps in retaining the bees for more time and improve fruit or seed set. So this was very, very clear why the male part is retained in the hermaphrodite flowers. Next, I move on to the conservation of bees, why we need to conserve bees. In the world, there are an estimated 295,383 species of flowering plants. Now, of this, 87.5% of flowering plants entirely or partially depend on flower visitors for successful seed set. Now, in India, we have 21,411 species of flowering plants, which is 7% of the world number, okay, total number of species. And of this, nearly 16,000 species actually require pollinating agents, so we cannot ignore pollinators. And 87 of the 115 global food crops depend upon insects for pollination. And according to Klein, 35% of global food production depends on pollinators. Okay, if the pollinators are not there, we are at a loss of nearly 35% of the food that is being produced. And annual economic value of pollination service worldwide is estimated to be around $577 billion. Okay, so there is a very recent paper. I think there's one more paper by Kala et al. In 2016, which says $577 billion. So do we give any value for pollinators? In fact, there is a very famous statement made by Pawan Sukhdev, who was with the United Nations, and he actually published a series of deep reports. They are the economics of ecosystems and biodiversity reports. And in one of his reports published in 2010, he makes a statement that not a single bee has ever sent you an invoice. And that is part of the problem, because most of what comes to us from nature is free, because it is not invoiced, because it is not priced, because it is not traded in markets. We tend to ignore it, and we take pollination for granted. We do not give any value for pollination because we do not pay for it. So that is the simplest statement that Pawan Sukhdev has made. Now, Basu's group from Calcutta University published a very interesting paper in 2011. In which they simply looked at the productivity of different kinds of crops. They classified the crops into all the cultivated crops in India into three categories. One is pollinator dependent crops, crops which depended highly on pollinators and crops which are independent of pollinators. They do not depend on pollinators like the cereals like rice, wheat, jowar, bajran, all these things. They are pollinator independent. And interestingly, they found that over the years of 1964 to 2008 or 2010, they found that there is a steady increase in the productivity of all those crops which were not depending on pollinators. It was mainly because of the efforts by the breeders. So they were year after year, they were finding new varieties, they were finding new genes for high yields, and they were actually releasing new varieties with higher yields. And that is how we have high yielding varieties of rice, wheat, and many other crops over the years, which are significantly different over the years. But in all those crops which are completely or partially dependent, highly or partially dependent on pollinators, there was a stagnation in this increase in the yield. The same kind of plant breeders are working on these crops also, but they have not been able to achieve much success with the increase in the yields. There is a kind of a stagnation in the increase in the yields, and they say that is it because these are dependent on pollinators and we are neglecting pollinators. Now maybe all these red gram and many pulses come under this category. Okay. Now supposing if we conserve pollinators, probably we can increase this also significantly. So that is what is Basu's idea. Now, how we have to justify? We can justify for conservation of pollinators. For this, we have to do what is called as economic valuation of pollination service. So economic valuation is done by identifying the crop and working out the total area under cultivation, the annual production of the crop, the area under cultivation in the entire region. We can take the entire country into consideration and the total production into consideration and the price per unit produce. And we have to work out the pollination dependency index, which is a very important thing. Now, how is pollination dependency index is worked out? We need a detailed study on pollination biology of the crop that we have selected. Then pollination efficiency of flower visitors. One most important thing that we should keep in mind is that all the flower visitors need not be pollinators. As I said, in case of pigeon bee, there are about 12 species of bees which are visiting, but of these, only one particular group of bees, 3 species may be efficient pollinators. Okay, all others, they are not pollinators. They may be stealing nectar, they may be stealing pollen, but they are not involved in pollination. So you have to identify which is the most efficient pollinator for conserving. So identify the most efficient pollinator, conduct exclusion experiments to determine the actual role of pollinators. So it is very important that exclusion experiments are conducted something like this. So you have the pigeon pea plot here where the pollinators are completely excluded by covering it with a shade net completely, not allowing any flower visitor inside, and then you have an open pollination system. You compare the yields and then see what is the difference. So any crop for that matter, it has to be, this kind of an experiment has to be worked out. So after the pollination, in the pollination experiments, you have with and without pollinators, set of plants to be enclosed in cages, a similar set left for open pollination, and you count the number of flowers produced in both the cases, number of fruits, pods, or seeds set in both the cases, and percent difference in set between the two treatments. So these are the observations that have to be recorded. And all this is expressed as a fraction of 1. So what will be the pollinator dependency index? If there is no difference between the two plots that you have, then the index is 0. That means the pollinators are not important. If there is only very little increase, like about 5 to 10% increase in the set with the pollinators, the index will be 0.05. And if there is a moderate fruit set, 25 to 40% set, it is 0.25, as it happens in case of red gram. If it is high, more than 50% set in the open pollinated system compared to closed system, it is 0.65. And if it is essential, that is more than 80% set, as in the case of muskmelon that I have given, it is 0.95. 0.95 is the highest index. It can be taken as 1 also, because there is no way in which there is self-pollination in many plants. So the productivity and production data from where you get area and production of the crop to be obtained from published sources. Like there are FAO reports, there are national production reports, there are ICAR reports, Indian Council of Agriculture research reports, different institutes publish reports. State Department of Agriculture it produces the reports, okay, publishes the reports, and because we should have a basis for this data. So the source of information that you have to give when you say that the annual production is so much, the area is so much, you should have the basis for that. And area is multiplied with productivity. That is used for working out the economic valuation. So it can be for an individual crop, a group of crops, a state, a zone or region, a country, or the entire world. Now, how it is done? The pollination deficiency index is PDI. Total value of the commodity is TVC. Total annual production is multiplied by the unit price for TVC. So the economic valuation of pollination service, EVP, is PDI multiplied by TVC. So that gives you the EVP. Now I'll give the case study of muskmelon. Total production is about 962,000 tons per year, and the unit price per ton is 32,000 rupees per ton. The pollination dependency is 0.95, PDI is 0.95, and the total value of production is 3,078 crores, the total value, and the value of the pollination service, it comes to 2,925 crores. So for the bees are responsible for this. So if you take the case study of pigeon pea, the total production is 37,50,000 tons, and the unit price is 50,000. This is all for the the entire country. Okay, the pollination dependency I've taken as 0.25 because there is also a possibility of self-pollination here. The total value of the production, it comes to 18,750 crores, and the value of pollination service, it comes to 4,687 crores. So you see that so much of value will be lost if the pollinator, if the megaphile species are not there. So much of production is lost, so much of money we will not be getting. Now, we have done this similar exercise for nearly 7 crops, okay, like cardamom, coffee, pigeon pea, muskmelon, bitter gourd, brinjal, and guava. And the total value of the pollination in these selected crops, it comes to about 27,800 crores. Okay, so which is unimaginable. Nobody will even imagine that bees are responsible for so much of income to our farmers or to the country. Okay, so that is the major thing. So working out economic value of pollination service is important, mainly for conserving pollinators, for conserving their nesting habits, for conserving biodiversity, and for convincing the policymakers. There is a very interesting, photograph. In fact, in June 2010, a food store in Rhode Island, it made a small study. As part of a campaign to highlight the importance of bees, temporarily they removed from its produce section all the food that depended on pollinators. So this is a picture of the market, and from this, the store from which they removed all those crops, all those products which was because of the pollinators. And they found that of the 453 items, 237 items vanished. That is more than 52% of the items they vanished. So they had to remove all these, and the store became almost empty. So this was a way of telling people why the pollinators are important. And Hannah Nordhaus, in one of her books in 2011, The Beekeeper's Lament, she says that bees are the glue that holds our agricultural system together. So they are the glue that hold our agricultural system together. If the bees are not there, probably we will not enjoy our breakfast, our lunch, or our dinner. Without bees, we can't do anything. So this is my team of students who worked with me, who continue to work with me. And thank you for listening.
Thank you, sir. Thank you.
Any questions? I'm ready to answer.
Yes.
Yeah.
This session is open for question and answer.
Yes.
Meanwhile, I have the
Yeah.
one question
Yes, sir.
related to
Yeah.
bees diversity.
Yeah.
In earlier mention about there is different type of family is included
Yes.
in
Yeah.
Apioda and specifically Epiformis. In that there is a Meletidae and another one family. Their number is very low in
Yeah.
India. What are the
Collected, yeah.
what are the reason behind it? There is possibility of this evolutionary numbers is not increases in the Indian
No,
population.
the possibility is maybe the collections have not been made much. Maybe they are very particular. I'm sure that if somebody starts working on these groups, he will definitely get more species. No, so that is also possible, and probably your answer also, your point also is important. Yeah.
Okay, okay. And another things there is stenotide bee is only getting from Australian region,
Yes. Yes.
not in Indian subcontinent or other part.
We don't have that. Yes. Yeah.
There is a that is also the related to topical and subtopical
True.
reason or
True.
maybe temperate
Yeah.
reasons possibilities behind it too.
We really do not know the reason for this kind of this one, but only for a few groups it has been worked out, mainly in the American continent, in South America, and such other places. Like for Meliponidae, they have worked out, and for Bombinae in the European region, they have worked out why they are restricted to certain regions. Probably the climate, also the environmental thing, and also the plants on which they depend. Also, all these things probably determine their distribution. Okay, so that is possible. Why certain groups are restricted to certain regions? Like for example, Coletidae in I think in the American continent you have hundreds of species, whereas in India we have only about 21 species or so. So that is the thing.
I think
Any
there
other
is,
questions?
in chat box there is no any questions seen. So, one question is there.
Yeah.
So, is it any alternative way to settle the honeybee in our modern farming field?
For honeybees, it is very easy in the model forms. You can do that. Honeybees, it is very easy, and the only thing is you should see the flora that is available. Somebody should work out a kind of a floral calendar in the area. What are the different species of plants that are there, and then when are they flowering over the year? From January to December, which are the species which are flowering, which are actually giving, which are attractive to the bees. Or you can also planting such plants and then introduce colonies. Okay, so that the bees are there throughout the year. And at the same time, you also can think of, as I was telling about bee hotels, you can think of keeping these kind of hollow tubes, okay, hollow wooden tubes. There are also ways in which in wooden, let's say, blocks, you can drill holes and then keep them in the field. So they come and then occupy these solitary bees, they occupy those, and then you can gradually increase their numbers.
Okay. And in the Vishwakarma, they asking that the alternative way for the close farming for the pollination.
Alternative way for?
Close farming. There is shade net is covered by the agriculture field. What are the alternative way?
Which plant? Which clove you said? Is it?
Close farming. There is shade
Close
net.
farming, yeah.
Design that time there how to design the pollination part.
Ramnath sir, can you read out the question that?
Yeah.
Question is that there is any alternative way to settle honeybee in our modern farming in close farming?
Yeah, you can. You can keep that in the. Again, you should know what is the kind of the plants that are there. Whether there are crops all through, or whether there will be any gaps. So in the gaps between the seasons, what food source that you can keep. In that case, you may have to artificially feed also. Okay, there are ways of artificially feeding the bees also. You can keep sugar solutions and then bees can survive. Yeah.
Sir, one more question. Actually, in our college, we set up 3 to 4 times the honey bees, means hives we were brought and try to culture them, rear them. But every time, like after 2-3 weeks, they start disappearing. So what can we do? Because we keep
There
the
can be two reasons for disappearing.
Yes.
One is too much of disturbance. Probably there is too much of disturbance nearby. Then the second thing is the availability of flora. So the right plants for the bees, you should see whether they are getting nectar and enough pollen to continue to stay. If they, supposing if you go to a place where there are no hotels at all, okay, you will not like to stay there, is it not? You'll go to some other town or some other city where there are some good hotels where they give some good food. So it is similar to that for the bees also. Yeah.
Okay, sir. Okay. Thank you so much, sir, for such a wonderful presentation as well as satisfactory answers. We all are very satisfied with your answers and presentations. Thank you, sir. And now the vote of thanks will be going to present by Ms. Harshada, madam. So over to you, madam.
Good afternoon, sir. Thank you so much, sir, for your great talk and presentation. I hope, sir, your guidance and the deep knowledge about the bee biology much more beneficial and informative to us and our students, that is UG students, PG students, and research students. And it will be help to students finding their opportunities in their career. So really, sir, you have a vast knowledge about that, and I'm very happy for that. And with permission from the principal, sir, Dr. V.B. Gaikwad, and our head of department, Dr. V.R. Kapute, sir, and coordinator, I declare this session is over. Thank you for everyone.
Thank you. Thank you very much. Thank you, Ramnath, for giving me this opportunity. Yeah.
Sir, we'll explain you more.
Continue contact in future also. Yes.
Yeah. We'll definitely come to you in future
Sure.
for such a lectures as well as hands-on trainings too related to the bill.
Okay. Yeah.
Thank you, sir. Thank you so much.
Thank you. Bye.
Bye, sir.
Bye.
Bye, sir. Dear participants, you need to wait for a few minutes more. Don't leave the meeting. Okay, so we have one announcement related to the feedback form and certificate. Tomorrow we'll share the feedback form link, and after filling the feedback form, you'll get the certificate. But most of you, if you try to feed the feedback form and it is like pop-up message will be there, what we can say, the link is blocked like that. If such a kind of messages are there, then don't worry. Next day, that is on day after tomorrow, you can retry it. The link will be open. So for the 3 days, we'll keep the link open for the feedback as well as certificates, but no need to write your data repeatedly, because we're going to restrict the response to one member only, and whatever name you want on the certificate, you put it there, right? So correctly you will write it everything in the feedback form. Accordingly you will get the certificate from tomorrow onwards for next 3 days. If you don't get the certificate on tomorrow, don't be panic. Okay, you can try on the next day. Okay, thank you so much for attending the 3 days national lecture series, and thank you so much for your cooperation, as well as we want you again during such a lecture series, as well as the webinars or workshops, conferences which we're going to attend in future. Even if you guys are also going to organize such a things, so let us inform. Okay, so we'll also join the same thing. Thank you so much for your cooperation. Now you can leave the meeting, all of us. Thank you.
Thank you, ma'am.
Yes, link. Thank you. Link will be shared on the Telegram group as well as through all the sources which you have received this link. We'll share to every group as well as on the Telegram too. Okay. Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thank you.